Hello, hello, hello. We are back. We are back. We are back. Today, boys and girls, we begin the seminal penultimate unit of the year, topic 14, molecular genetics. And bringing us in is one of the best bands ever made. And that is REM with one of their best tunes called Losing My Religion from their hit album Out of Time. Run, run, and get this album and listen to the whole thing. Not a bad song in the bunch. Gotta turn it off now. All right? A little molecular genetics. So we took a look at the history of genetics, the history of heredity and Mendelian genetics. This is modern genetics. Modern genetics, the core content, basically speaks to what is called, excuse me the cough, the central dogma of molecular biology. And the central dogma of molecular biology is the fact that DNA is transcribed onto RNA, which is then translated into making a protein, And you know that it is all about protein. It is all about protein. All right. And so, but you can see the three things on this slide that we are going to be taking a look at in this topic particularly. How is DNA replicated? Because we kind of know how it is made during the S phase of mitosis, but how is it replicated during that phase specifically? We'll take a look at it. And then 2 and 3 are the process which we call protein synthesis, which is kind of actually called transcription and translation. The two of them together make up what is called the central dogma of biology. First, though, we need to go back into November of 2019, before any of us were wearing masks and seeing each other over computer screens. When we were sitting in the classroom, and you know what we were doing in the classroom there? Hanging out. Girls were talking to the girls. Diego and Barrett hanging. Ollie doing whatever Ollie does, you know? And now we're here, all right? And we're going to take a quick look. I'm not going back. What you got is what you got here. All right, let's take a look. We're going to review DNA versus RNA. Do you remember this molecule? Well, you should. All right, so this is a DNA molecule. This is a portion of a DNA molecule, which we call a nucleotide. Remember, there are 4 classifications of macromolecule. We have, all right, can you name them? Follow along. Carbohydrate, protein, fat, and what you are looking at, a portion of a nucleic acid. All right, the smallest unit of a nucleic acid, the glucose, the amino acid of a nucleic acid is a nucleotide, and you are looking at it right here. All right, so we want to take a look at the nucleotide. Again, has 3 components, a 5-carbon pentose sugar, which is B, and that sugar is a form of ribose. Okay? A phosphate group, PO4. Let's put that little negative closer. It's a negatively charged molecule, the phosphate portion. And a nitrogenous base, which is always drawn. So you have the circle, the pentagon, and the kind of rectangle. This is a simple diagram that gives you basic information about a nucleotide. This is going to be brought up and put into your Cornell notebooks as part of topic 14, 13, 14, whatever topic we're on here. All right. In this, but we do know it's the second to last topic of our year together. So both the phosphate group and the nitrogenous base are attached to, all right, so let's go over something. Let's do a little honors biology. I'm not sure if we did this way back in November, but if not, we'll do it again. Remember, you have your pentose sugar, and we start off here at the point of the pentose with an oxygen, and then every point is a carbon. Every point is a carbon. I'm going to put the pen on for this one because it might be a little easier for me to actually write with this then. All right. Let's get this pen. Good. All right. Right there you have carbon number 1. You have carbon number 2 right here. The next point, carbon number 3. This point, carbon number 4. Just go back in your Cornells and look. And if you did and carbon number 5 will be up here. All right, I'm going to put a quick circle around carbon number, okay? number 5, number 3, and number 1. All right, super important. Not the carbon number 2 and 4 aren't important, but they're just not as important to us. All right, why? Because carbon number 1, when we look at the DNA molecule, carbon number 1 has, all right, that's why I put a line here. Carbon number 1, take that off there, has a nucleotide, let's make this a little smaller, has the nucleotide attached to it, has the nucleotide attached to it. So it's carbon number 1 on both, all right, has the nucleotide attached. Carbon number 3 and 5, okay, basically attach. Get out of there. Attach sugar to phosphate, basically the wrong. Sugar to phosphate, so important. So if you're holding on to a ladder and you think of DNA as a ladder, right? Think of DNA as a ladder. Think along this way. What you hold on to as you climb the ladder, that is number 5 and number 3. What is what you step on? The nucleotide bases, number 1 to number 1. So it makes it super important if you are trying to figure out how to draw a DNA molecule, as in what you see on the next slide here. This is a DNA molecule, all right? So we take a look, I'm going to erase that, I don't know what that is, but let's get, I don't remember from last year what that is. So if you take a look here, and you will notice the DNA molecule from what you remember from earlier in the year, the structure of DNA. What is it that you want to know? All right, so the nucleotides, all right, notice they connect to each other, and they connect to each other, ladies and gentlemen, by hydrogen bonds. All right, we'll go over what the A, T, and G, C means again in just a second, but for remembrance now, is that between the bases A and T, there are 2 hydrogen bonds, okay? And the C and G bases, 3 hydrogen bonds, 2 and 3, and a hydrogen bond. For remembrance from your biochemistry, put this into your notes. The weakest kind of bond, strongest bond covalent, followed by ionic, followed by hydrogen, because it is a hydrogen bond that must break in order for DNA to replicate, just like the hydrogen bonds break when water is changing phases. Weak bond, it's a weak bond. All right, so in order for the bases, look at the entire molecule. So I'm going to take away the erase. I'm going to erase here. So this way we have it. Look at the entire molecule. All right, you're looking at it? You're looking at it? Okay, in order for the bases to be facing each other, the A and T and G and C, all right, and thus able to pair, the strands of DNA must be running in opposite directions. Look at the O on the left-hand strand and the O on the right-hand strand. It looks upside down. Well, that's because in the picture it is. Okay, so the 2 DNA strands are described as anti-parallel. Did I spell parallel right? Did not. Is that spelled right? No, it's not. I'll just do this. Right, my spelling is off today. Anti-parallel. All right. Yeah. See, if I had somebody here like Jay Blatter telling me the spelling, I'm name dropping from the year, because I miss you all. It's anti-parallel. What does anti-parallel mean? Okay, this is what anti-parallel means. Look at it. Anti-parallel means the DNA strands are running in opposite directions in order for the bases. Why? Let's go back one. Because DNA can only bond at carbon number 1. And if you had 2 strands written like that, there's your carbon number 1, and there is your carbon number 1. You see it? You got to really pay attention here. Be engaged. Some of you are not engaged. Be engaged. All right, anti-parallel. That's a little bit of honors biology. And as the anti-parallel strands lengthen, the DNA will organize themselves into a stable configuration. All right, but how do we notice and how do we test that in science? Let's say, how do you realize which side is which? They go by numbers, and it really is super easy for the person or persons who invented how we show it in tests, the antiparallel structure. When we put little numbers, the left-hand strand, you have you have your 5 strand and your 3. On the other side, opposite, your 3 to 5. And that's how you see it on a test. And how is that named? And who named that? Well, on a DNA strand, if you're looking here, whenever the DNA strand ends on one side, there must be a free number 3. Notice how it bonds to make the ladder that you hold to. A 5 and a 3, a 5 and a 3, 5 and a 3, 5 and a 3. You get to the end, you got a free 5 and a free 3. On the other side, it runs in the opposite direction. So that's the way you can tell on a test. If you are given the 5 to 3 end, you've got to fill it in with the 3 to 5 end. All right. So that, because replication is what it's about. All right. Replication is what it's about. All right. So anti-parallel, the new concept here, understanding what 5, 3, 3 to 5 mean. And they usually also denote it with a prime, meaning an apostrophe. The science calls it a prime sign, 5 to 3. 3 to 5. So it's an apostrophe after it. It's a small thing, but it's important, especially for questions that I'm going to be posing to you here. All right. The DNA nucleotides, let's review those quickly. There are 4 of them. All right. An adenine, thymine, guanine, and cytosine. And you might remember earlier in the year when that question made it onto your test, and I said, no, don't count it because we didn't cover it yet. Well, we're going to cover it right now. All right. So you will notice A and G and C and T are categorized as either a purine and a pyrimidine. All right. Let's think big picture. And this year, with big picture, you get to IB, we'll get a little more together. Big picture, it's the structure. All right. There are 2 fused rings in the purines, adenine and guanine, and there is 1 ring for the pyrimidines. That's all I want you to be able to know this year, to recognize 2 versus 1 and then categorize the 2 of them. So here we have the phosphate, the sugar, and the base. And you'll notice when we pair them up, as you saw on the last panel, a purine and the pyrimidine always pair together, ATGC. So bring out the pen again. You have your two pairs, your AT, and you have your GC, your AT and your GC. And both of those together form what we call the double helix. All right. The DNA has 2 strands. Eventually, this strand is going to twist like this to form the double helix. The strands are always held together by hydrogen bonds with an A pairing with a T and a C pairing with a G. That is called Chargaff's base pair rule, which is another thing that I'm going to send you off to take a look at on your own. These scientists, it's good learning. Give them some credit. An A with a T and a G with a C. You twist this up, looks sort of like that, and you have the double helix. The 2 DNA strands are anti-parallel to each other. All right. Again, this really is the basis of modern, at least honors biology here. All right. We'll go on for one more, the base pair rule. 2 hydrogen bonds with A and T, 3 hydrogen bonds with G and C. Here you can see a little bit more of a twisting. Notice this diagram, though, doesn't give you the 2 versus 3. So it's not the best diagram there. All right. ATGC.